Hello, this talk aims to introduce the basic ideas in physics that sparked the technological development of two medical imaging modalities, magnetic resonance imaging, or MRI, and positron emission tomography, or PET. This will be a possibly pleasurable itinerary across the momentous discoveries of the early 20th century and the visitation of a few of the main protagonists of that period. This journey will end with a short equation that gave theoretical predictions about A, the existence of particle spins, and B, the positron, without which both MRI and PET would not exist. Now, every lecture on this type of physics always starts with a picture, actually the picture of Albert Einstein. And since this is a lecture of a novel YouTube channel, it would have been nice to do something really different. Hey, but we didn't. One cannot really avoid starting with a picture of Albert Einstein, because modern physics started with Albert Einstein. In particular, special relativity and the ensuing disruption of the old idea of time dominated theoretical and experimental work in the following years, taking the field to uncharted territories, to great discoveries, and to exciting technological developments. But what is it, special relativity? To understand it, we will ask for the help of two very good friends, one standing on a moving ledge, while the other one is on the ground watching. One more thing, although they are good friends, they are in big disagreement. Since they are so close and yet so angry with each other, they must be Italian. So we will call them Mario on the ledge and Luigi on the ground. But why are they in such a disagreement? Mario is looking at the two lasers in positions A and B, and he thinks that they are shooting rays of light at the same time because they reach him at the same time. Luigi strongly disagrees. For him, the ledge is moving and the laser in A has to fire first. Otherwise, how could he reach Mario, who is moving away? Remember, nothing can move faster than the speed of light, so the two rays must travel at the same speed. As it happens, special relativity explains it by saying that all is relative, including time. So they are both correct. Simultaneity depends on the observer. So Mario and Luigi now can get on with their lives, happy again. Special relativity as a scientific idea was so profound, so different, that all physical models of entities and particles moving at high speed had to be changed. In particular, take the simplest and most important model of all, the model of hydrogen. You will surely have seen this model before, say in secondary school. Hydrogen has a central nucleus with one proton with a positive charge, a neutron and one electron in an orbit. This simple model was first presented by Bohr to explain some experimental findings, such as the spectrum of light when it goes through hydrogen gas, and this spectrum presented neat bands of absorption. Truth is, however, that the clear and simple model of Bohr was already not working very well. Importantly, it could not explain why a charged particle, the electron, could stay in an orbit around a nucleus of opposite charge with the proton without crashing into the nucleus itself. Now, de Broglie, a French physicist, explained that. He postulated the wave nature of the electron and showed that the reason why the electron was not getting away from its orbits and crashing was that these orbits were the only orbits that allowed a standing wave. However, the Bohr's model did not account for relativity effects, and it was down to an English physicist, Paul Dirac, to sort this problem out. He did it with very elegant mathematics that, in the end, collapse into one very short equation, the Dirac equation. By doing so, he described the quantum wave functions that now added to de Broglie's model the relativistic effects everybody was looking for. Let's now pause for a moment and focus on the character, Paul Dirac, the man. Even his closest friends, and admittedly he did not have many, called him the strangest man. His personality quite likely belonged to what we would call now the autistic spectrum disorder. At total ease in the world of mathematics, atoms, spins, and so forth, he was clearly not much interested in human interaction. I think you can easily pick him up in this group picture taken at Max Born lab in Göttingen, where clearly the paper he was reading that day could not be put down, not even for a minute, to smile at the camera. The social issue, however, was a major problem for Dirac, but not just for him only. This picture shows Paul Dirac with Heisenberg and Schrödinger arriving at Stockholm train station, bound to receive the Nobel Prize for Physics in 1933. While Schrödinger was accompanied by his wife, both Dirac and Heisenberg took their moms to the prize presentation. But eventually, as this photo shows, Paul Dirac did meet a woman, Margit Wigner. Nobody could explain at the time how he managed to start a conversation with her. However, initial communication between the two took place. It never really ended, as really she did pretty much all the talking for him ever since. They eventually married and went on to form a very happy and successful family. Let's go back to the equation now. The equation described the orbits in a way that matched experimental data quite accurately. However, it also made two startling predictions. The first one was that charged particles had a spin, meaning they were rotating around their own axis. As they are charged, and as any moving charged particle generates a magnetic field, that meant charged particles such as protons generated a magnetic moment. That is to say that the magnetic field had a certain strength and a direction. The magnetic momentum of water protons is the basic principle behind the MRI, as we will call it from now on. Water protons have a magnetic moment that generally is directed randomly, resulting in null overall magnetization. However, when a strong magnetic field is switched on, the spins will align to the direction of that magnetic field and will spin with a rotating speed that will be dependent on the intensity of that magnetic field. Hence, the first main component of an MRI scanner is a strong magnetic field. The second important component of an MRI scanner is gradient coils. These are magnetic fields that change linearly along the X, Y, and Z axis, so that each point in the field of view has a slightly different magnetization, hence a different spinning frequency for the protons. Now, the third component of an MRI scanner is a radiofrequency coil that emits pulses. These pulses will excite the protons as long as their frequency of excitation is the same as the one of the rotating spins. This excitation pulse will send the spins into a different state, so that the net magnetization resulting from all the spins will form an angle of, say, 90 or 180 degrees with the direction of the original strong magnetic field. The fourth component is a radiofrequency detector, although sometimes the same coil can work at both excitation and detection. That once the pulse is completed, we'll start listening to the radiofrequency signals emitted by the protons. Note that each spin in a different location will send a signal with a very specific radiofrequency. Now, once the pulse is switched off, the water protons will go back to their initial state, which means that their magnetization will decay back to what it was before, parallel to the magnet. Now, the rate of decay of the net magnetization depends on the tissue the water protons are in. Hence, by collecting these magnetic decays at different frequencies, which correspond to different locations, a computer program will generate maps of magnetic relaxations, what we call MRI images. This is a demonstration of the exquisite anatomical detail that can be achieved with this technique. MRI images of the brain can nicely differentiate structures and tissues with very high resolution and great signal-to-noise ratio. However, the imaging potential of MRI extends quite a lot further than using water as a contrast agent between tissues. Water moves in directions that are constrained by the structure of the tissues. In particular, the quite elongated shape of neuronal axons pushes water across specific paths. By following the signal emitted by water molecules, one can construct a three-dimensional representation of brain axonal paths. The rendering of these paths can be achieved with so much detail that such images can be easily confused with pathological specimens. In these techniques produce the same or higher level of detail than post-mortem tissue dissections. But MRI can do imaging also without water. One straightforward application of MRI is the use of paramagnetic agents, such as gadolinium, that stay in the vasculature once injected and enable the visualization of brain vascular branches. Now, water protons are not the only ones to give out a magnetic resonance. Other molecules, such as creatinine, choline, N-acetyl aspartate, as shown in the picture, produce a resonance, and these can be explored using MRI radiofrequency signals to explore not space, but just choosing a big voxel in the brain, and exploring the different molecular resonances that can be used to quantify the molecular milieu of the brain. Now, one striking advance in MRI applications has been the discovery of the so-called Bohr effect, where Bohr stands for blood oxygenation level dependent signal. The Bohr effect depends on the paramagnetic properties of hemoglobin. Hemoglobin is the protein in blood that brings around oxygen to the peripheral tissues, where oxygen is then released. Oxygen-bound hemoglobin or not changes its paramagnetic properties. Hence, difference in oxygenation can be easily imaged with MRI. Since oxygen consumption increases when a piece of brain tissue is particularly active, we can use the Bohr effect to visualize brain functional activity when a subject is performing a task in the MRI scanner. All we have seen so far, MRI, stemmed from one only prediction from Paul Dirac's equation, the spin. But there was another one. One solution to the equation was, as we mentioned before, the electron and its trajectories. But another solution popped up, a solution that had the same mass of the electron but opposite charge, a positron. This was totally unheard of, as a positron would be an element of antimatter. No one had actually ever seen antimatter before, so this was a very daring and strange prediction. What is extraordinary is that the positron was actually found a few years later in Carl Anderson's lab at Caltech. The group was measuring cosmic radiation using gas chambers. The picture on the left here shows the gas chamber with a polarized bar. The bar will bend the trajectory of a polarized particle, whereas the bend will depend on the mass of the particle. The picture shows the trajectory of an electron-like particle, but the bending is in the opposite direction of the one expected by a negatively charged particle. The picture was kept in the files for a while until they re-looked at it and realized that what they were observing was not electrons. It was a positron, antimatter. There is something unique about the positron. Now, it's generated by a nuclear reaction by proton-rich nuclei. Once generated, it survives a little after the expulsion from the nucleus. It is slowed down by interactions with other particles until, after generally say a few millimeters from 1 to 3, it finds a slow-moving electron and it annihilates with it, emitting 2 gamma photons. Gamma photons have a lot more energy than traditional X-rays that are generated generally just by electron transitions. And this is the cool thing. Electron-positron annihilation is the only known reaction whereas mass is entirely converted in energy. This makes for positron or positron emitter atoms to be great energy containers. But if you were a Star Trek fan, you would know that already. The spaceship Enterprise indeed had positron batteries to sustain its trips to the edge of the universe. Positron emitter isotopes such as oxygen-15, carbon-11 or fluorine-18 can now be produced by local cyclotrons and can be attached to any substrate, say a drug, sugars, and then injected into humans. The concentration of the radioisotopes will be small, but because it can pack so much energy, it can be visualized by a ring of detectors that stop gamma radiation. And this is PET, or positron emission tomography. With positron emission tomography, we can visualize the biodistribution of almost any substance and measure its kinetics across tissues, obtaining very valuable functional information in vivo. In this example, a sugar analog was labeled with the positron emitter fluorine 18, allowing a three-dimensional visualization of glucose uptake in the whole body, and, notably, in the brain, that we know is the only organ with a glucidic-only metabolism. And all this is the heritage of Dirac equation. Thank you for listening.